get started. So hello, everyone, and welcome to this week's Paleo Perks. We'll have Thomas Clements from the University of Birmingham talking about taphonomy with a talk titled All Fossils Are Rotten. Understanding decay is key to understanding exceptionally preserved fossils. But before we get to Thomas's talk, we'll just do a quick logistical notes. So if you've never been before, our format for the seminars is this quick welcome and announcements. Thomas will give a 30-minute talk. We'll do a question and answer and then move into small discussions using breakout rooms. And then after that, Thomas has agreed to stick around to talk with us for a little bit in our tea time following the breakout rooms. So if you have questions during the talk that you'd like to be included during the question and answer session, please send them via chat to the questions at Paleo Perks host, who's Elizabeth this week. So more housekeeping. Paleo Perks values the participation of everyone interested in paleosciences. So please remember to abide by our code of conduct during today's seminar. You agreed to it when you signed up to get the links. If you found yourself here without signing up, please do head to our website and sign up so you can get the emails and check out the code of conduct. Please keep yourself muted for the duration of the talk until we get to the breakout rooms and the tea time. Again, you can ask questions by using the chat feature and send the questions to the Paleo Perks questions host. And that's also if you're having technical issues where you should send those as well. We are accepting nominations, including self-nominations for our potential speaker pool. Again, that link is at our website. If you think of anyone that you think would give a really great talk, please let us know, including if that person is you. We also, a new feature fairly recently for our Zoom platform is closed captioning built into Zoom. You should see in your Zoom controls a little button that says CC, and you can use that to show them or hide them. There's also a transcript available if that's something that would help you to type a little bit easier. And then finally, we have a optional survey monkey that we'll throw into the chat that just collects sort of demographic info. It's anonymous and optional, but encouraged. And if you can just help us get an idea of who's watching. So without further ado, today's speaker is Thomas Clements. He got a bachelor's at the University of Brighton, a master's at Bristol, and then a PhD at Leicester, and is now a fellow at Birmingham University. And he is going to again talk to us today about taphonomy. So Thomas, if you want to go ahead and take the screen share.
Okie dokie. All right. Let's hope this doesn't break this time.
Perfect.
Excellent. Can you see that? Is that all right?
Yep, looks good to me.
Wonderful. Okay. Hello, everyone. My name is Thomas, and I'm going to talk to you today about why all fossils are rotten and about why we need to understand decay in order to understand exceptionally preserved fossils. And to do that, I'm going to give you a little run through very, very quickly about taphonomy, a crash course in taphonomy, and then I'm going to talk to you about some of the experiments that I've been doing as part of my research. So taphonomy, the word taphonomy is this, what it means is the study of the processes that affects an organism after death that potentially results in fossilization. And taphonomy comes from the Greek. Taphos means to burial, and onomy is obviously the study of, so it's the study of burial. And taphonomy covers a lot of different subtopics. So everything from an organism when it's alive, there's what we would consider biology. Then you have necrology, so that's the study of how an organism dies. Then you have biostratonomy, which is what happens to an animal's carcass after death. Is it scavenged? Is it transported for hundreds of miles by a river? Is it ripped apart by currents? That sort of thing. And then the part of the fossilization process, which I'm most interested in, is diagenesis. So that's what happens after burial and the geological processes that turn organics into rock. And then you have a fossil at the end, hopefully, fingers crossed. And the part of the talk that I'm going to be talking about today is a little bit of everything. And you're going to get a smattering of all three things. So why is decay such a big deal? Well, the simple answer is that in nature, everything is recycled. You may have heard the phrase that there are two truths in life, death and taxes, but in fact, there are three truths, which is death, taxes, and the fact that you're going to rot when you die. We can stop rotting in some ways, or we can slow it down by using our fridge, but decay is observable around us all the time. Stuff like your bananas going brown and black and becoming horrible. The fact that when you open your door in the morning, you don't have to wade through a sea of dead bumblebees. These are because nature is naturally recycling everything all the time when organics die. So what is it that breaks down organics? Well, organics are broken down by scavengers and detritivores. So classic scavengers are things like vultures, and detritivores are things like mushrooms and things like that that eat sort of rotting matter. But mainly the thing that I'm interested in is bacterial metabolism and this really cool process called autolysis, which is actually where your cells, once they die, they can't maintain all the structures inside them, and your own enzymes start eating your own cells from the inside out. So you basically chew yourself apart. It's a pretty fascinating subject, and I could do a whole talk on cellular autolysis, but those are the main things that break you up when you're dying and rotting. And one thing that's really important to talk about is that decay happens everywhere all the time, and it happens when you're buried. So there's this weird idea in paleontology. Sometimes people think that if you bury something, it's going to turn into a fossil, and that simply isn't true. So in this case, our little dinosaur has been buried and its soft tissues have decayed, but then its bone continues to be decayed and until it's completely obliterated. And nature is really good at this. And it's really, really good at recycling. It might take a long time, but it will eventually get rid of all of the bones. They will recycle. That is, unless a process occurs to stabilize the organics. So in this example, the process is permineralization, and that results in the skeleton becoming a fossil. And we're all pretty au fait, I would hope, with dinosaur fossils. But the important thing to note here is that this is not fossilization, this is preservation. This process is preservation. And that's because fossilization is one outcome of a range of processes that affect organics after death. The opposite of fossilization is non-fossilization, and that is the norm. So 99.9% of all living things ever that die will never turn into a fossil. There's just no chance of that happening. There's a very, very small percentage that turn into fossils. And fossilization, as I said, is all the processes. So in this example, it was everything that happened from the minute the animal died all the way up. That is what fossilization is. So paleontologists sometimes conflate preservation and fossilization, but they are two very separate things. And one thing that we need to note is that as organics move from the biosphere into the lithosphere, they lose information. And that's really important because for paleontologists, that's a big problem. So how is that information lost? Well, basically, it's because organics are broken down based on how easy they are to be eaten by the bacteria. So generally, it's much more energy efficient for bacteria to break down the squishy bits than the hard bits. So that's what they tend to go for first. So we tend to lose those bits quicker. Oops, for some reason, my PowerPoint is now not turning. There we go. So, but even within squishy bits, there is a sequence, and this sequence isn't random. Experiments by Sanson et al. in 2010 showed that this isn't a random sequence. Generally, you get things like the brain or the lungs. This is an example, by the way, but things that are not very resistant to decay, they will tend to disappear before things that are more resistant to decay. So for instance, in this example, the intestines and the stomach are more resistant to decay than the brain is. And that's the order that generally the bacteria will sort of remove information from the fossil record, or from animals, what I should say. However, there are some places in geological time that have environments that allow or disrupt this stripping of information. And these sites are called Lagerstätten, and that comes from the German word, the storage place, to store. So in our example here, our little dinosaur has been buried and then has started to decay, but a process, in this case mineralization, has occurred, which has stopped all of the soft tissues from becoming lost, and also permineralization has occurred on the skeleton. So in this fossilization process, we have biomineralized information retained, but we also have some soft tissue information retained. And that's really useful. This would be a gold mine for paleontologists because much more anatomical information is preserved. Soft tissues are really vital because they give us much more information about ancient animals. They allow us to reconstruct the animal more accurately. They allow us to fit them into phylogenies, into family trees. They allow us to in some cases, they allow organisms like this squid, which is predominantly a bag of water, to actually turn into a fossil, which is a very rare event. But also, these sites are much more representative of ancient ecosystems, so they're better windows into the pasts. So they're really, really interesting, and paleontologists or taphonomists are really interested in trying to understand what environmental conditions allowed this preservation to occur, because we would like to understand that. Now, most Lagerstätten have shared characteristics, things like low levels of oxygen, low temperatures, maybe high salinity, rapid burial. These things all tend to act to prevent or slow down, or as we would say, retard the decay of organic material. But one thing that's important to note is that every Lagerstätten is unique environmentally. There are no two Lagerstätten that are the same. And that's because there are so many factors, so many environmental factors that are happening during the preservation process. And mineralization is the main process that basically prevents organics from being removed or destroyed. It's not the only process. There are other processes, but mineralization is the main one. It's what I'm going to focus on today. And much like ice cream, mineralization comes in many flavors. So you can have silica that preserves things, or pyrite, or clay templating, or calcium phosphate, which is going to be the main crux of what I'm going to talk about today. So basically, processes like mineralization will disrupt information loss. So you might get fossils where most of the internal organs are lost, but things like the muscles are preserved, and normally muscles would rot away very quickly. And you find those muscles being preserved because they've been mineralized alongside biomineralized tissues. Now, sometimes these processes act on specific soft tissues and not on hard tissues. So we get these really weird fossils where we get soft tissues being preserved, but not hard tissues. One example that I like to dredge up is Rollins akita, which is a beautiful fossil polychaete from the Cretaceous of Lebanon. And in this polychaete, we have muscles being preserved, but all of the hard parts of the polychaete, such as the bristles and the jaw and other hard parts, they're all lost. So a lot of paleontologists have looked at these kind of things and said, well, this doesn't conform to what we understand about decay. And so therefore something weird is happening here. And this is a weird fossil that sort of goes against everything that we know about decay. And I'm going to talk a little bit about this fossil later on. Another thing that happens that's a bit weird during preservation is that we get very specific minerals will seem to target specific organs. And that can lead to some pretty weird and wacky fossils as well. And one example that's really famous is in the Cambrian. We get a lot of these weird arthropods that are being preserved with things like brains, and these brains are preserved in a mineral called pyrite. And this can lead to some weird and wacky ideas about how these brains are preserved, because the rest of the organs and the soft tissues and the muscles are lost. So something very bizarre must be happening here. And this can become really, really confusing from a paleontological perspective when you have multiple soft tissues being preserved in multiple modes of preservation. So that is different types of mineral acting on different organs. You can get some really bizarre and wacky fossils happening, and can sometimes appear like preservation is random. And we're going to talk a little bit about that as we go through the talk. So how do we make sense of these weird and wacky fossils? Well, the answer is experimental taphonomy, and that's what I do. So the TLDR, the easiest way of describing what I do, is that I create very simplistic experiments, like as simple as possible with the fewest possible variables. And I identify a fossil that I'm really interested in. I try to find a modern analog. So in this case, this is a fossil lamprey at the top of the screen. And then I've used a modern lamprey, and we decay that modern lamprey, and we look at we can map the normal sequence of decay for the modern lamprey, and then we can use that sequence of decay on the fossil to try to determine how far the fossil has deviated away. And if it has deviated, maybe we can try to work out what's causing that. I should just point out at this point that I did not rot this lamprey. This was work done by my PhD supervisor, Professor Sarah Gabbett. And another thing that I should point out at this point is that one criticism that taphonomists often get is, why do you use simple experiments? Why do you not try to recreate ancient environments? And there are two reasons for that. The first reason is it's impossible. It's impossible to know the exact environmental conditions of ancient environments. But also, it wouldn't actually be that informative, because we're interested in the overarching processes, not the processes that are occurring necessarily at one specific site in one specific point in geological time. But we can do that, but we'll talk about that in a bit. And another thing that I need to get across early doors is that, really bizarrely, even though the bacteria are digesting all the organics and removing the organics, they actually drive mineralization. And that's really important, because then we need to understand the timing of mineral replacement. And that's one of the most important things, because preservation is this weird balancing act. You need the bacteria to eat the organics to start mineralization. But if they eat too much of the organics too quickly, then nothing is going to be replaced. So as taphonomists, we try to understand the balance between those two things. So it's experiment time. Let's talk a little bit about some of my experiments. So the first one, I'm going to talk to you about some taphonomic biases that occur within an organism. So this thing that I'm going to talk about happens in a lot of different fossils, in fossil sites all around the world. But I'm going to talk about fossil fish from the Cretaceous of Brazil in Santana. And these fossil fish are amazing fossils, but when you CT scan them, they're even more amazing, because inside them we have beautifully preserved internal organs. So we have things like the gills that are pink, we have the heart, which is red, which you can see the individual valves in. It's pretty amazing. We have muscles in red, and then we have the intestines in orange. And all of these are preserved in the mineral calcium phosphate. But the interesting thing here is, why do we get muscles and intestines, but no gonads or no kidneys or no liver in this particular fish? And we see that pattern consistently happening in the fossil record. We have this weird selective preservation occurring. So one thing I need to very quickly talk about with calcium phosphate is that we know a lot about calcium phosphate. We've done, I say we, but lots of this was done before I started researching. But scientists, paleontologists have a really good understanding of what it is that allows or controls calcium phosphate from replacing soft tissues, what it is that actually causes it. And the answer is that calcium phosphate needs an acidic condition. So this is pH along the side here, the numbers, and pH is basically a scale of acidity or alkalinity. So at the top, 14, we have drain cleaner, which is very alkaline, and 0 is incredibly acidic, not something you'd want to spill on yourself. And then pH 8 is seawater, so it's just above neutral, it's slightly alkaline. And that's really important, because in seawater, calcium carbonate preferentially precipitates, which is a lot of words to basically say that normally rocks in seawater formed from calcium carbonate. And calcium carbonate is really cool. You can get fossils in calcium carbonate, but it will not replicate soft tissues in the same way that calcium phosphate does. The calcium phosphate requires an acidic condition, so it needs a pH below 6.38, so below normal seawater. And paleontologists have identified that when you rot a carcass, or when a carcass rots, it generates this acidic, basically bubble of fluid around itself. So normally this would be in sediment, but in this example, I'm putting it in a little jar. And it creates that acidic environment because the bacteria are respiring while they're eating it. And also, like all of we talked about autolysis earlier, all the acids that are leaking out of you, all the enzymes are very acidic. And as the bacteria are metabolizing, they're basically releasing lots of carbon dioxide, which mixes with the water and creates carbonic acid. So basically it's creating a little acidic bubble, lovely acidic bubble, perfect for calcium phosphate. But this raises a very interesting question. Why is it then that we get selective preservation inside the fish? Why do some organs preserve and some don't if we're creating this acidic environment? Well, one hypothesis is that the internal organs create unique microenvironments, whereas the counter or the null hypothesis is that all of the organs are inside a giant, basically inside the internal cavity. There is this basically little soup that's very acidic, and all the organs are sitting in that soup. And if that is the case, if the null hypothesis is correct, that means that something else must be controlling phosphatization. And I was really interested in trying to understand that, so I did some experiments. So I got a fish, and I identified 3 of the main organs: the stomach and the intestines. We commonly find those phosphatized in the fossil record, but we don't find the liver commonly phosphatized. In fact, we very rarely find the liver at all. So what I did is I got these fish, I made tiny little incisions inside of the flesh, and then I basically inserted pH probes into the organs so that I could map the acidity of the organs as they were decaying through time. And then I sealed up those wounds so that it was almost as if the fish was just decaying normally. And then you can plot it on a graph like this. We have pH running from alkali at the top to acid at the bottom. And we have this dark gray window at the bottom. Basically, if the pH drops inside that, then calcium phosphate can precipitate. And that's called the calcium carbonate phosphate threshold. It's a bit long-winded, so we'll just say 6.38 from now on. So a fish looks like this, normally. Within about 30 days, it's looking pretty ropey. And by 60 days, it's literally just slime, bones, and fat, and a few of the fins. So the reason why I show this is because this is happening very quickly. This is not on, decay does not happen on geological timescales. This happens really quickly. So if we log the external conditions, you can see that this bubble of acidity forms around the fish. That's great. We kind of expected that to happen. Now, if I show you all 3 different internal organs very quickly, we have the stomach, the intestines, and the liver. And if we plot their averages, all of them are well within inside the calcium phosphate window. And what's interesting here is that all of the organs are within the window, including the liver, which was slightly surprising. So we're not seeing unique microenvironments. And what's interesting here as well is that we don't see any sort of weird changes in between all of them. They're all sort of doing the same thing, much lower than the external conditions. And if we look at what happens inside a fish in real time, we can see that in less than 5 days, the organs look pretty bad and not looking great. Within 10 days, the organs are almost completely gone. You can hardly recognize any of them anymore. You can recognize the intestines, but that's about it. And by about 15 days, there's no organs left. So what that's telling us is that for phosphatization to occur, you need to have this acidic conditions, but it also has to happen very, very quickly. It's not operating on geological timescales. So this is a bit of a messy slide, but basically we've got the hypothesis with the microenvironments at the top and the null hypothesis at the bottom, and our data shows the null hypothesis is correct. So all of the organs are sitting within this soup that's acidic inside the carcass. So why is it then that some of these organs are preferentially phosphatizing and others are not? Well, the answer is really simple. Once you enter that threshold, the acidic threshold, the organs that tend to phosphatize are also the organs that have the highest phosphate content in their tissue histology. So basically, it's a tiered bias. Once you enter the phosphate window, you have to have lots of phosphate in order to basically phosphatize. So the reason that we don't find many livers phosphatized in the fossil record is because they don't have much phosphate in them compared to muscles and the stomach and the intestines. So the take-home message from that little experiment was that phosphatization happens early and it happens very specifically, and it acts on tissues with high phosphate content once you've already met the criteria of the acidic conditions. And that everything else, once phosphatization has occurred, everything else continues to rot. So you can start to get these weird fossils, a bit like this fossil worm, where the muscles that are rich in phosphate have phosphatized, but everything else that isn't phosphate rich continues to decay absolutely normally, doesn't get mineralized. And so you get these weird fossils that almost seem to be counterintuitive to a normal decay sequence. Right, so that's the first experiment. The second experiment I'm going to talk to you about is a bias between 2 related taxa. And we're still going to be talking about calcium phosphate. And we're going to talk about my favorite animals, which are the cephalopods. So we're going to talk about octopuses and squid. basically, these two animals are related. There's hardly any differences between them, really. The only way to really tell the difference between them is based on the number of limbs they have. So octopuses have 8 arms, squids have 8 arms and 2 tentacles. But biologically, they're very similar, and they are very closely related. But for paleontologists, what's really interesting is that we find lots of fossil octopuses all preserved in calcium phosphate. So again, this is from the Cretaceous of the Lebanon. We get these beautiful fossil octopuses, but we don't find any fossil squids. And that's really weird because the molecular clock data and also fossil evidence, so they do have some hard parts, like they have little arm hooks and they have little beaks. And those beaks and arm hooks are found in the same deposits together. So we know that they diverged around the same time in the Permian, but yet we don't find any fossil squids. So why is that? So there's only one thing for a taphonomist to do, and that's create an experiment. So I got some squids and some octopuses, and I decayed them. It's very disgusting work. Every experiment I talk about here is horrible. It stinks. It's very unpleasant, very slimy. The smell gets in your hair. So when you go for a shower that evening, it all comes out. Sometimes the animals explode. But if you want to know about that, you can ask me questions later. But basically, we can plot it on a graph again like this. It's the same deal, pH on the left, time on moving along the right. And if the pH falls inside the gray window, then phosphatization is likely to happen. So you can see there's 2 lines. One does not enter the phosphate window, and one definitely does. It'll be no surprise to you that squids do not ever enter the calcium phosphate window. And in fact, within 15 days, they completely rot and fall apart. They're just mush. Whereas octopuses are the opposite. They actually stay together. They're very robust. And their pH of their body tissues falls inside the calcium phosphate window really rather rapidly. So that means that they're much more likely to turn into phosphate. So this had us scratching our heads a little bit. It makes sense why we don't see phosphatized squids, but why? Why is that happening? And in order to answer that, we had a look at their ecology. So they have 2 very different modes of life. Squids like to swim. They sit in they hang in the water column, whereas octopuses live on the seabed. Now, swimming is very energy expensive. If you're constantly swimming all the time, you're burning energy all the time. So cephalopods have evolved a really clever evolutionary trick in that they use a metabolic waste product. We have it too. When we pee and we poo, we excrete ammonia. Now, cephalopods also have this ammonia, but squids have evolved to keep that ammonia within their tissues. And that's really important because ammonia is lighter than water, so it floats. So they basically just float in the water column and they don't have to keep swimming all the time. Octopuses don't want to float, so they excrete all their ammonia and get rid of it. Now, this is really important because if I show you this diagram again, if I just remind you that you need an acidity below 6.38 in order to turn into calcium phosphate, in order for your soft tissues to be replaced, ammonia has a pH of 11. This means that the ammonia in the squid tissues is buffering their pH. So it's preventing them from entering the calcium phosphate window and turning into a beautiful fossil, which is a bit sad, really. But we just to test this, we made sure we investigated the tissues, and we saw that the tissues in squids have much higher ammonia than in the octopuses and also in fish, which were our controls. So this shows that there's a taphonomic bias between these 2 animals caused just by their evolutionary choices, if you can choose your evolutionary choice. So squids have basically evolved a really neat trick, which has prevented them from ever turning into a fossil, which is kind of sad in a weird way. So these 2 animals, even though they're related and very closely related, have a very different preservation potential, which is very surprising when you look at them and consider that they're both basically just bags of water. Cool. So I've rushed through those 2 experiments. Last experiment I'm going to talk through is a taphonomic bias based on paleogeography. And we're not going to talk about calcium phosphate. We're going to talk about something a little bit different. This is close to my heart because I did my PhD on the Mason Creek Lagerstätte, which is a 300 million year old fossil bed. It's found in Illinois. And it's an amazing fossil bed because you get beautifully preserved soft tissued animals preserved inside these hard rock concretions. They're sort of this sort of size of this beautiful orange rock called iron carbonate or siderite. We call them siderite. And you can see this beautifully preserved fossil worm here. And I was really interested in the fossil worms because we find quite a lot of fossil polychaetes or hard parts of polychaetes in the fossil record, but we very rarely find their soft tissue. So I was really interested in these little critters. And one thing I noticed when I was doing my PhD is that we saw a huge amount of variation, even within one species of polychaete in the same fossil bed. So all within the Maison Creek. This is a bit of a weird diagram, but stick with me. So in the rows, we have a species of polychaete. So in the top row, we have Escanites. In the middle row, we have Diodonticasta. And in the bottom row, we have a Streptoscolex. And all of the animals that are labeled 1 on the left, they're what I would say are they're the really good, well-preserved polychaetes. And they've got all of the morphological characters. They're like, they could basically crawl away if they weren't long dead. The ones in the middle are kind of like, they're okay. They're not badly preserved, but they're not as well preserved as the other ones. And then the ones on the right, in the right column, they're the real meh fossils. They're like, you can hardly tell what they are. You can only basically work out what species they are based on the shape of their jaws, on the morphology of their jaws. So I was really interested in why we see this variation within one fossil bed. And it led me to this question: is the variation in these polychaetes random? And that's a really important question, because if we would expect that within one fossil bed, all the fossils are undergoing the same processes of fossilization. They're all undergoing the same mineralization processes. So why do we see such variation? And is it random? Now, polychaetes are awesome animals, and they're really useful for decay experiments for a couple of reasons. They're really good analogs because A, we know loads about their anatomy. B, they've got a really good mix of hard and soft characters. So they've got really hard jaws and these little chaetae, these hairs that pop out of it. That's why they're called polychaetes, because they've got these hairs. They also have, they're really cheap. You can get hold of them really easily. And they have a really good fossil record that goes all the way back to the Cambrian. They haven't really changed much since the Cambrian. So what I did is I looked at a huge number of fossil polychaetes, and I basically catalogued all of their characters that we would expect to see based on modern analogs. And some of them we never see because of the type of preservation, but that's fine. And then what I did is I basically catalogued them. So each column is an individual fossil. And what I did is I scored the characters. So 0 means those characters are not seen, and 1 is that the character is seen. And then I catalogued the color is based on the mode of preservation. And this is really important because this allows us to quantifiably compare huge numbers of fossils and actually like mathematically say this fossil is better preserved than this fossil. And then I can compare that data to fossil decay data. And there's been loads of experiments on polychaetes, rotting them. And one thing that's really important to note here is that decay, the sequence of decay, as I talked about earlier, is not random. So we see the same patterns of decay consistently every time you rot a polychaete. And what we found was that characters that decay quickly in our experiments on modern polychaetes are rarely seen in fossils. And characters that decay slowly in our polychaetes are commonly seen in fossils. Now, it sounds really obvious, but this is the first time that we've ever been able to mathematically show that, like statistically show that in the fossil record. And what that means is that not only is the sequence the same across different taxa, if they're related, but also it means that the variation that we see is not random. It's basically that these animals are undergoing different amounts of decay prior to fossilization. So why? Why is that happening? Well, the answer is paleogeography. When we think of a fossil bed, we often think of a point on a map, but the Maison Creek is a 300 square kilometer fossil site. It's massive. And the paleo environment is basically a marine basin that's very close to a very large river. And the simple fact is that when you're close to the river mouth, there's loads of deposition of sediment and minerals. And if you're far away, there is very little. So what that means is if you're an animal and you live close to the shoreline, you get buried very quickly when you die, and there's lots of minerals. And if you live far away, you're rotting before you're being fully buried, and you're also probably more likely to be scavenged. And we see that in the fossils. The fossils do confirm that. So in conclusion, what we see here is that the variation in the Maison Creek preservation is not random, but it's a combination of burial speed, mineral abundance, and amount of decay. And that even though this variation can seem random, it does actually conform with experimental models and is predictable. We can actually use our models to model what is going to happen taphonomically based on these geographical sort of biases, if you will. So what are the take-home messages of this talk, other than taphonomists talk really quickly and rocket through experiments far too quickly? Well, basically, if you're a paleontologist, your fossils are rotten. Simple as that. And that taphonomic biases can occur within an individual. They can also occur on a taxonomic level and on a geographic scale. So basically, taphonomy is messing with your fossils no matter which way you look at it. And you need to think about these kind of things no matter how you use the fossil record. So if you're describing fossils, especially soft tissue fossils, be very careful not to overinterpret, but also at the same time, try to be aware of the taphonomic story or the taphonomic influences on your story. And also, if you're doing large-scale paleontological work like diversity data and things like that, then your data is almost certainly skewed by taphonomy. Another take-home message is that understanding preservation and fossilization is stinky, horrible work. But my big take-home message is that mineralization and soft tissue preservation is not random, but it sometimes is so complex that it can seem random. But that hypothesis-driven experiments are basically key to understanding this mineralization and key for understanding soft-tissued fossils. So just to round things off, I'd like to leave you with a quote from Richard Leakey, who said that when out fossil hunting, it's very easy to forget that rather than telling you how a creature lived, the remains actually indicate only how they became fossilized. And that's not strictly true, but I think within a taphonomic context, it's really important to remember. So thank you very much for listening to my talk. I hope I didn't go through it too quickly, and I'm very happy to answer some questions and then chat to you guys afterwards if you have any questions or interests in learning about horrible stinking experiments.
Yay. So thanks. That was a really fun talk. Lots of very cool dead things. And we've already got some questions coming in. So I think around the time that you were finishing up the second experiment with the squids, Roy asked, what are the implications of the squid results for lack of ammonoid soft tissue preservation?
That is a really great question, and a question very close to my heart. So basically, it has every implication. So squids and octopuses derive from a 10-armed ancestor, and that 10-armed ancestor itself will have branched off from somewhere deep in the cephalopod family tree. And almost certainly ammonites probably split off from that branch at some point as well. Now, obviously, we can't be sure about ammonites because we don't know anything about their soft tissues, but there is a high probability based on looking at other cephalopod groups and using sort of phylogenetic bracketing that this trick that squids use to sequester ammonia is probably not super derived. It looks like octopuses have developed the skill to basically get rid of it and not keep that tissue, not keep that ammonia within their tissues. So it's highly likely that the reason we don't find any ammonite soft parts is because they probably had lots of ammonia in their soft tissues as well. But we don't know. That's kind of guesswork. There are other taphonomic factors with ammonites that you need to take into account. So for instance, the fact that they have this buoyant shell probably meant that unless they somehow got buried very quickly, they probably floated in the water column rotting, and their body parts probably fell out long before they ever got buried and long before they sank to the bottom. Now, there is one example of a potential ammonite stomach crop. If that is correct, and that is a stomach crop, then that indicates that an ammonite, when it does get buried, isn't likely to phosphatize, because we do find other phosphatized fossils in that fossil bed, which would suggest that our hypothesis about the ammonia is correct. However, there's a lot of speculation about whether that is an ammonite stomach crop. So I think the easiest answer is I don't know. The long-winded answer is we suspect it could be quite similar. I'll shrug.
So a sort of follow-up. Are there other living groups of pelagic organisms that have high levels of ammonia that impacts their preservation potential? Like maybe sharks or freshwater fish or?
Yeah, so sharks is a good one. And my supervisor, who actually supervised that project, is Danish. And when we were doing the experiment, he was telling me all about the, I can't remember what the name is. And even if I did know the name, I'd probably butcher the pronunciation, so I probably wouldn't say it. But in the Scandinavian countries, they bury sharks. I think in Iceland, they bury the sharks and they let them ferment. And he said that the overriding smell you get is ammonia. Now, that would make sense because we do find some phosphatized shark remains, but they're very few and far between. And actually, if I'm being 100% honest, I've never actually studied them. And I haven't looked at modern shark tissues either. But it's something that at the time when we were thinking about this, it was something that we wanted to expand the project into, to look at shark tissues. But it's not something that I've done. But I think it's highly likely, yeah, you probably bias the fossil record because ammonia is used by quite a lot of marine organisms as a buoyancy control, basically, because it's easy to, like, we all produce it, so it's easy to sequester one way or another. But yeah, that's a great question. Good project to do, I think.
So we've got another one that I think went through to everyone, but I'll read it out loud. Would these acidic microenvironments also affect negatively certain types of tissues? For example, carbonate-rich tissues like bones being totally or partially dissolved in specific regions around or within these microenvironments?
So no is the short answer, because if you want to dissolve bone, you need some pretty intense acid. You need to drop down in the pH really significantly. We're talking like pH 3, 2, 1. And some pretty nasty acids. And the acidity generated by decaying organics is no way near enough to dissolve bone away. Now, there are some caveats that I should say, because I think in one of my slides, I showed that you can have mineralization occurring early, but then you can have other processes which might stop things like bones being preserved. So you get these weird fossils that have soft parts but no hard parts, which is really counterintuitive. Sometimes in some sediments, we can have environmental conditions that are so acidic. Think of like things like peat bogs and things like that. They actually can denature and then eventually bones will dissolve. We can have those. And if mineralization occurs on some tissues early, but the environment is generally unfavorable to bone preservation, they can dissolve away. And actually the Maison Creek is a really good example of that. So we get this soft tissue preservation, but then the bones continue to decay away and they just leave hollows, they just leave imprints. So there's no actual bone material left in them, which is really weird. And I think it made a lot of early paleontologists really struggle to understand and reconcile why soft tissues that are prone to decay seem to last, whereas tissues like bone that are really hard that should survive didn't seem to turn into fossils in those fossils. And I think that's that can happen, yeah, and it can create some really bizarre looking fossils that are very sort of confusing and weird, if that makes sense. And actually has created a lot of controversy within taphonomy, because in the last few years there have been a very small group of people on the fringes of taphonomy who have sort of said, well, actually, when you do a decay experiment in water and you compare that to the fossils are really different from what we see on the decay sequence. And so therefore your decay sequences aren't marrying up to what we see in the fossils. But the simple answer is, if your fossil is deviating from the expected decay sequence, that's telling you that environmentally something weird was going on, not that the fossil was not conforming to not that the organism was not conforming to the normal decay sequence. So actually that's a really great question, because if you do see that weird dichotomy of no hard parts but soft parts, that's telling you that environmentally something was weird. Or once the fossil formed, then you've got like weathering processes or something diagenetic that's happened way after the fossil formed that's being weird. So it's very confusing, because I've only talked about what happens immediately after the animal dies and in early diagenesis. And I haven't even, in this talk, I didn't even talk about the weird weathering stuff that happens a long time after and can strip out loads of fossil information. Yeah.
All right, so I think we've got about 2 questions left. One just came through also to everyone. What would be the best proxy to recognize a vertebrate in the fossil record, and what would be the first vertebrate currently for you? Because, sorry, it was cut off, because there is a random decay in soft tissues.
Yeah, oh my goodness. Wow. Well, I think first thing, whoever asked that question is basically put me out as a sacrificial lamb, because no matter how I answer that, I'm going to upset someone somewhere. So thank you for such a difficult question. I'm going to be a bit of a coward and say I don't work on enigmatic early vertebrates because, for exactly this reason, I don't like the controversy. But I mean, animals like Pacaya, from the Burgess Shale and places like that are very strong contenders and would make sense because taphonomically speaking, we know that places like the Burgess Shale and the Chenjiang and places like that do preserve soft tissues. So we know that those organisms existed. But I don't think I would feel comfortable going on record saying what I think is the earliest, not even vertebrate, but chordate. So I don't know how to answer that question. And there are lots of taphonomists who do work on that sort of questions, and they get very angry at each other. And so I'm not going to tread down that path. But I don't know what a good proxy would be either. It's so hard to know, isn't it? Because there are so many weird enigmatic creatures, and that's a big problem with taphonomy, that we can rot modern analogs, and for some groups like hagfish and lampreys, we have modern analogs, but for a lot of these enigmatic, deep time animals, we don't have any modern analogs, so we have to use what we perceive as the closest relatives, which often are hagfish and lampreys, but that can cause some difficulties. So great question. Come and argue with me later on, and we'll chit chat about it.
So we keep getting questions in, but maybe just a couple more. So another one, you mentioned about the decay experiment in your talk and compared the decay rate of different organs. I wonder whether this is the results you obtained solely from your experiment on fish. Did you test this organ decay experiment on other animals besides fish and polychaetes? Great
question. So the answer is that I was not the first person to do this. There's an amazing paper by Rob Sanson that came out in 2010. And they looked at it in hagfish and lampreys, especially. And they were the pioneers of this. And within hagfish and lampreys, they showed that the sequence of decay is consistent. Every time you rot a hagfish, it happens pretty much the same way. Now, there are some caveats there. Like if you chopped a lamprey in half and then decayed it, it will decay slightly differently than if you decay the whole lamprey, because bacteria can get into the injuries and they can start eating away in different ways. So there are some caveats to that. But in fish, we see my experiments in fish that it was consistent, even across different taxa. So I rotted several marine fish, like things like mackerel, sea bass, sprats, things like that. And then also some freshwater fish. And we do see a consistent pattern. And actually what's really interesting as well is that I didn't do this work, but police and people like that have done experiments on human cadavers, and the human cadavers map in a very similar way to the fish. So it's, by process of elimination and guesswork, we can probably deduce that although the pattern might be different between major groups, like generally as a rule of thumb, like a brain will go before an eyeball, which will go before skin, which will go before bone, and so on.
All right, so we're going to do one last one and then let you have a break for a little bit. So this will be the last question for the question and answer, but stick around for the tea time, and we've got a really good one for a fun tea time opener. So I wondered about the effects of lipid content of various organs on decay rates / mineralization potential. The liver is a relatively fatty organ, certainly in land mammals and birds compared to other organs.
Great question. Yeah, so I actually wrote a grant just before I got my fellowship on investigating that exact question. So I have no answer for you, but stay tuned, and maybe in the future I will be able to tell you the answer. But almost certainly, tissue histology has massive implications. So in my experiment, like what I talked about here was a very simplified, dumbed down version, but basically, so in my results here, I talked about how the muscles have more phosphate in them than the liver. So they're more likely to become phosphatized. But there's loads of other factors as well that I didn't even get into. So for instance, if you have more cellulose in your histology, phosphate, calcium phosphate is basically more likely to bind to certain things, certain biological histologies than others. So muscles, are full of ATP, they're full of phosphate, they're also full of collagen, and collagen is like a key binding site for phosphate. So calcium phosphate preferentially bonds to that collagen. So if you're made of collagen, you're much more likely to phosphatize than something that's not made of collagen. Now, lipids have some do some really interesting things, because lipids, not only do they have like weird chemistries, they're also quite weird to break down. Like bacteria need quite special techniques to break lipids down, especially really long chain lipids. And so there's so many weird questions about like what impact lipids have. And it's something that I'm really interested in and I'd like to work a lot on. And no one in taphonomy is really doing that right now. No one is looking at like specific tissue histologies and trying to break these things down and looking at like composition. Like for instance, when we're looking at the phosphate content, we were having to use papers from like the 50s and 60s, and they're really old papers because just no one has looked at it in such a long time. So we had to go away and like look at it ourselves. So it's really tough. But I think lipids is a really good question. And there are so many other factors, like also things like electrical charges. Like different organs have different like electrostatic charges depending on the structure of the tissue, and that can massively influence binding sites for minerals. So there's it's so complex. Like what I presented here was really dumbed down, but yeah, absolutely, lipids is probably a factor. I don't think it's a controlling factor for phosphatization. But I do think it's definitely a controlling factor for a lot of the others. I wouldn't be surprised if it wasn't a controlling factor for maybe pyritization or something like that. But truth is, I just don't know. And it's something I want to work on in the future.
All right. Thanks so much again for a really great talk. We'll head into the breakout rooms in just a second. I'm going to take the screen share to do the last few slides. So thanks everyone for coming to Positive Talk today. Again, another plug for the Survey Monkey that we'll throw back into the chat. And please come next week to hear Kostov Thiramalai from the University of Arizona present on methane monsoons and modulation of millennial scale variability. So we'll pop into some quick breakout rooms where you can chat for a few minutes with other paleo people. And as our suggested icebreaker for the week is, if you were to be fossilized, what mode of preservation would you like to have? And yeah, so you'll get an invitation to come to breakout rooms. If you're not really feeling breakout rooms today, stick around for the